I think it's time to start. Let's get going. You got me again. Today and tomorrow I'm giving my last two lectures, and they're both going to be about evolution in spatially structured populations. And today I'm going to go over more of the basics, and tomorrow I'll tell you about a few more things, including a little bit more about what I do sometimes. Let me just introduce the notion of spatially structured populations and think just at a high level why we care about these things. Individuals of a species are spread over space, and that space is sometimes heterogeneous. And in fact, that's what the context I'll focus on is when there are more or less discrete populations separated by inhospitable habitat, but then another population somewhere else where the organisms of the species we're thinking about can live. And this is just a landscape, I think, of northern Wisconsin, showing you all these different habitat types, and you can see that most of those are quite heterogeneous and separated. There's barriers for dispersal on the landscape, etc. And here's just a couple of other places. Here's where I come from, and here's where we are. This is essentially an ecology map of British Columbia. These are different ecological zones. We got a lot of mountain ranges in BC, and you can see how fragmented that landscape is. And that's not in this case caused by human activity. That's caused by the way geology happened in BC. And there's, you can see, just a lot of heterogeneity. Species that live on mountaintops are separated from other mountaintops by valleys between. Species that live in rivers are separated from other rivers by the dry land. Same for lakes. Same for patches of forest. Same for patches of grassland or desert, etc. There's a lot of spatial heterogeneity in nature, and species live in this patchwork of places. And then, of course, India is similarly heterogeneous, especially as you get up toward the Himalayas. Okay, so this spatial heterogeneity of suitable habitat is a fact of life. It's true for almost every species, and even the ones which live in a more continuous scale. Space is still a factor because individuals tend to interact with other individuals nearby more than they do individuals further away, and that has consequences as well. Now, I'm going to, as I said, today I'm going to start with some introductory stuff, and I'm going to show you some things based on some very, very simple models, which is the right place to start. But before I go there and pretend that the world is simple, I just wanted to show you a few examples of the way the real world has heterogeneity and deviations from those models, because that'll be a theme a bit today. So here's just a few different structured populations, chosen either because I just got the picture somewhere. This again is northern Wisconsin. This is the Åland Islands in Finland, where Ilkka Hanski has done a lot of work on the metapopulation structure of the fritillary butterflies, the Granville fritillary. These are fort fungus beetle. Well, these are fungus. That little black dot that you can't see is a fort fungus beetle. This is what I did my PhD work on, actually a really long time ago. So these fungi are the food and the habitat of the beetles. They live on dead or fallen logs. The beetles require this fungus or two other kinds of fungus that also live on fallen logs. And the intervening spaces between logs are not suitable habitat for the beetles. There's nothing for them to eat there. They tend to stay on these logs for most of their life, and some occasionally move between them. So you have, even on a fine scale, a population structure. And then this picture down here is, this is actually my son from 11 years ago. Mainly I put it in here because I like showing a picture of my son to you. He looks a little bit different now. And he still likes her. But I show you the picture because humans, of course, are also a habitat for a lot of a lot of other organisms. And if you've ever had a child in daycare, you know that, well, we're habitat in particular for a lot of pathogens, diseases, etc. But we're not uniformly afflicted by these diseases. And the population structure, the specifics of the spatial structure of those diseases matters quite a bit. If you've ever had a child in daycare, you know that daycares are these sources of colds and viruses and other diseases that the kids then bring home and infect you, and you're sick constantly for the first 3 years of having your child in daycare. The kids somehow seem to not get them, but they convey them to you. So you have a source population of the viruses and then the sink populations of the parrots and the adults that get them. And of course, there's a lot more tragic and more important dynamics of diseases in a spatial context than that, but they're not as cute. So I'll show you that. Anyway, so metapopulations are everywhere. Sorry, I should define this term. I won't actually use it again much, maybe. Metapopulation is a word used in a couple different ways. I use it here just in the sense of a collection of populations, a population of populations. So a species that's subdivided into several different local populations. Sometimes people imply with the word metapopulation that some of those populations occasionally go extinct. That seems to me an unnecessary distinction because every kind of species that is divided into populations, they will eventually go extinct. So a metapopulation, drawing a distinction between a collection of populations with extinction and without seems a bit pointless since that's the way they all are at one level or another. Okay, so why do we care about space? Well, we care about spatial population structure because it's out there, right? We care about it because sometimes the population genetics of space allow us to understand other features of species that we care about, understanding the demographic history of the species or understanding the dispersal between populations. Even if you don't care about the genetics might help you get there to learn about those things. But from an evolutionary biology perspective, the spatial population structure is important in a lot of ways, and they all share in common this, I think, this common feature, which is this, that when you have space, when you have a population, a species spread over space, and when you have limited dispersal, so that the individuals in that species are not moving freely over the entire range, then you're going to have the feature that, in general, individuals which are spatially close to each other are going to be more similar to each other. Okay? They're going to be more similar in terms of their genetics, okay? Because they're likely to have ancestors in common because individuals don't move that far, and that's what we'll spend a lot of time talking about today. So there'll be genetic, some diffuse genetic relatedness, perhaps, between individuals that are nearby. That causes them to have higher rates of homozygosity. It causes some other interesting features. Because those individuals are related, and because individuals which are nearby are the ones that are affecting each other in other ways, that means that individuals are competing with relatives, they're mating with relatives, they're interacting with relatives. And we've heard a little bit about spatial games from some of the late afternoon talks. The space matters in terms of the nature of who interacts with who, and I did it out of order, but the other thing is individuals which are nearby also tend to share features of their habitat, features of their environment. So individuals which are spatially close are more likely to be similar to each other in the kinds of selection pressures that they experience. Okay. So when you have this feature that basically we're generating covariances across the population of the kinds of genes and the kinds of selective constraints that individuals experience. Okay. So that's, in some sense, the root of all the interesting stuff about spatial structure. Okay, so there's a lot of different ways to divide up why do we care, what do we want to talk about, et cetera. And here's the kind of rough outline of what I'm going to try to do. When we think about population structure, as I said on the previous slide, that under certain circumstances that generates this feature that individuals which are nearby are more genetically similar. Well, how does that happen? In what ways do individuals who are close to each other become more different? Or phrased equivalently, why do individuals in different places become different from each other? Why is there variance among populations? Okay, so that's largely what we'll focus on today, is why do we get differences between populations over space? And then secondly, what are the consequences of that spatial differentiation on the way evolution works? Okay, so that'll largely be what I talk about tomorrow, right? Next time. Okay, so let's go in on that. Okay, so just keeping this at a very high level, conceptual level. If we think about it, what causes populations to get different from each other? What evolutionary forces make populations different? Well, a perhaps relatively minor thing in some species is that mutations, by necessity, occur in a single individual. Right? You might get the same mutation in somebody else, but any given mutation occurs in a single individual, and that individual has a place in space. So once the mutation appears, that place that it is becomes genetically somewhat different from somewhere else. And then that might be propagated by later drift or selection. Right? So that's in some sense the core of all variation. You have to have mutation first. So that's a minor force causing differentiation. But more important, or at least more important in the way we normally think about this, is the other two features here. If you have spatially heterogeneous selection, so if some alleles are favored in one place and another allele is favored somewhere else, that by itself is going to cause differences between those populations, genetic differences. Right? So allele increases in frequency in one place, decreases in frequency in another place because of selection. That causes those two populations to become more different. Right? Just very straightforward. The other thing that causes populations to diverge genetically is genetic drift. Right? So if the populations are different, if you have, to a large extent, individuals breeding with each other locally, then genetic drift can, again, by chance, it's drift, by chance, increase the frequencies of a particular allele in some places and decrease the frequency of that allele in other places. That leads to genetic differentiation. Okay? So those are our two biggies, selection, drift, that cause populations to become more different from each other. Okay? Now, drift that was able to proceed uninhibited by migration would lead to fixation of one allele in one place and another allele in a different place, and you could, in principle, get complete differentiation, same with selection. Okay, but I said if you had no migration, what homogenizes population, again, a lot of things can. Homogeneous selection can, so if you're selecting for the same allele everywhere, that can cause the populations to remain similar to each other, at least at that locus and perhaps nearby loci. But the big player in this, the one that affects the whole genome, is dispersal between populations. Okay, so we tend in the population genetics world to refer to this as migration. Okay, that's just a bad habit that we got from 75 years ago, and we keep doing it. But we're going to 85 years ago. We're to call this movement from one place to another migration. Now, more properly, to a good biologist, migration implies something like what birds do in winter and summer, what monarch butterflies do in some complicated way over North America, where the individuals in the population move someplace for one season and move back in a different season. That's not what we're talking about here. Okay? We're talking about the semi-permanent movement of individuals from one population to another, and then they tend to remain there. And if they move and actually successfully reproduce in that area, we can also refer to that as gene flow, right? So the flow of genes from one place to another. So there's a lot of steps in that, right? Individuals have to leave somewhere, they have to survive, they have to arrive somewhere, and they have to successfully reproduce there to count in the dispersal models that we're talking about here, right? So there's a lot of complications, fun complications mechanistically about how that happens. But we're talking about genes moving from one place to another because the individuals that carry them move and then reproduce. Okay? So that's the big player causing homogenization, right? So dispersal is like mixing. It is mixing of the populations. So if you have two distinct populations, one's fixed for one allele, one's fixed for another allele, and some individuals move back and forth between them, after a while they're going to be genetically the same, right? They're just mixing those two populations. It might be slow if migration rate's low, but they will eventually, all else being equal, become genetically uniform. Right. So those are the players. We'll put them together. Okay. There's a lot of different. All right, so I'm going to shift gears a little bit. We're talking about genetic differences between populations. We can characterize those in a lot of ways. We could just list the allele frequencies of some locus we cared about for all the populations and just look at that and try to tell a story based on other biology that we knew. We could measure perhaps the difference in allele frequency between a wet habitat and a dry habitat, something like that. That could be useful in certain circumstances. But a kind of general way that we often use to talk about the amount of differentiation between populations is something that we call FST. There's a lot of complications with FST in terms of how you estimate it, et cetera. And I'm going to say John's going to talk about it, but he's going to shake his head madly. No, he's not listening. No, no. Okay. But here's this FST. I'm going to go back to the basics on this. This is an idea that Sewell Wright came up with quite a while ago. It's a very useful concept. So just there are many different ways that people have defined FST. In fact, in the first paper, Wright essentially defined it in two ways, and I'll show you those two ways. But there's many other ways that people talk about FST. There's a lot of confusion about whether we're referring to it as a population quantity, as a parameter, or we're using it as an estimate. We don't have good habits about distinguishing between the estimates and the parameters, et cetera. Be very careful when you hear people using FST if it matters to you, to trace back exactly what they mean by it. But I'm going to show you a meaning of FST that goes back to the root, and it's a very useful one. So one way to define FST is this: that we take the list of allele frequencies in all the different populations. Okay? So we have P is an allele frequency, and imagine that's a vector, a list of the allele frequencies for population 1, the allele frequency for population 2, for population 3, all your populations. And you just take the variance of those allele frequencies. Okay? And then on the bottom of this is P bar. That's the mean of that same list. And 1 minus P bar. Okay? So the variance in allele frequency divided by this function of the mean, P bar 1 minus P bar, sometimes P bar Q bar, because we like to make it shorter. But that's the definition, right? So the more variance in allele frequency between populations that there is, the higher the FST would be. Okay. So if all the populations are genetically the same, what's FST going to be? 0, right? Great. And it turns out that when the populations are maximally different, with every population has a different allele, FST would be 1. Okay. Did all that. Any questions? Yeah. Hi. Yeah, that's an excellent point, and that's a fantastic point. So, and I'll talk about it briefly later, but not in this context. So, thank you for bringing it up. So, if migration or dispersal can be more complicated than what I've implied here, this is dispersal, kind of random dispersal with respect to the habitat or to the genetic features of the individuals. But you're very right that dispersal can sometimes have some condition dependence to it, so that individuals move to places that they are most comfortable or have the highest fitness, or for that matter, who have some other feature that they identify. So like I said, I'll come back to it later, but this is good now. When individuals move to a place for which they will have a high fitness, And you can imagine them doing that because they get there and they go, oh, this is the right temperature, or thank goodness there's food here that I can eat. A lot of ways that individuals can assess their environment. If they tend to move to places that are good for them, we call that habitat selection. And that can cause, as you say, a lot of differentiation between populations. Yeah. Thank you for bringing that up. Yeah. Yeah, the denominator is proportional to the variance of if you mixed everything together, what would the variance be? Yeah, and that's right. Another way to think about FST is that if we want to know, let's see, I'm going to do this for haploids first. That's another one of these pesky things where haploids and diploids matter. The rest of the day I'll talk about diploids, but in this case it's easier to. So if you want to talk about the variance among populations of the genetic variance among populations, for haploids, that's FST times what the genetic variance would have been in a panmictic population with the same allele frequency. Okay, so yeah, you can think of that over there as pulling that over. With diploids, just so it won't confuse you if you ever see it again. With diploids, there's actually 2 FST, and that's because there's 2 copies of each allele, and they're correlated with each other, and that causes this extra 2 to appear. But that's painful. The other feature, while we're talking about this, the genetic variance within populations is actually, and this is both diploids and haploids, same equation, is 1 minus FST times what that genetic variance would be in a well-mixed population. Okay, so it's a feature of thinking of the bottom as a population. Now, one of the things about FST, and this is actually a good bridge into that, one of the nice things about FST is that, so here I'm defining it in terms of the allele frequencies of a particular locus, right? So that might be, a particular, on average, p bar might be 0.137, whatever, right? And another locus might have an allele frequency of 0.521, something, right? So those are going to behave quite differently in terms of how much variance there is among populations. But on average, they're going to behave the same in terms of FST. This division by p bar or q bar winds up standardizing for the effect of the allele frequency. Okay, and you can kind of see that more, from these kind of equations. Whatever that genetic variance is, the structuring of the population is predictable from FST. Now, there's stochasticity around that. We'll talk about tomorrow, but on average, that's what we'd expect. We expect the same thing for loci that are evolving in the same way in the same populations, regardless of the allele frequency. Yeah. Nope. So with diploids, it turns out that there's actually more variance in the population. Sorry, the question was, shouldn't there be a 2 here, right? No, if you add these up, you get 1 plus FST VG. Yeah, there's more variation, and the reason is for diploids that you basically are getting a correlation, a covariance of. Oh, you're right. Brian's smart, but he hurts himself. Yeah, so the reason is there's a covariance of the alleles in the same individuals, and so if you're in a population with a high frequency of the big A allele, the other allele is likely to also be big A, and so that covariance of the alleles causes the individuals to vary more, and to get more variance among individuals than with diploids in a structured population. That's right. But yeah, there's no covariance. You can't define it because there's only one of them. Yeah. Yeah. Oh yeah. So if it's gender biased, and if you're talking about autosomes, there's not a big effect. You can use your these things, well, the equations that I'll show you in a bit, work out without big deviations. If you're talking about sex chromosomes or mitochondria, of course, in a species where mitochondria is maternally inherited, the male dispersal matters nothing. They're not moving. They're not causing gene flow of mitochondria. They're moving mitochondria, but then they're a dead end. Okay? And so then the sex bias dispersal matters completely. All you care about is the female dispersal. And with sex chromosomes, you could derive or has been derived what the right equations are, depending on whether you're talking about Y or X or what. Right. Okay. All right. So just rule of thumb wise, just if you're not familiar with FST, in many species this can be quite small, close to 0, 1 in 1,000, something like that. For within species comparisons, it tends to be in the range 0 to maybe 0.15, maybe 0.2. Quite a lot of species, it's smaller than that, 0.05 or so. That's the kind of numbers to have in mind when you're thinking about what these FSTs might be. Varies a lot among species. There's not an FST, but just so you have a ballpark to think about. Okay, so FST helps us understand how different populations are genetically. Now, so far, this is just a measure, right? We haven't done anything that talks about why this difference is there, right? This is just, here's a way we can measure how much differences there are. Now I have skirted that a bit. So with a constant amount of drift, same effective population size, and the same dispersal rate for different loci. On average, they should have the same FST. If you had selection on one of those loci and not the others, then they're not going to behave in the same evolutionary way, of course. So I'd say that to make sure that I'm not overstating. Okay, so here's another interpretation of FST. It's also a very useful one in a different context. And that is the FST is also a measure of the proportion of reduction of the heterozygosity of a population that results from the population structure. Okay, so we can measure how much less, or we can predict how much less heterozygosity we should see in a population as a result of the fact that they have population structure. Now, why would we have a reduction in heterozygosity? The reason is we don't have random mating when you have population structure and limited dispersal. Right? Individuals are more likely, with limited dispersal, you're more likely to mate with individuals that are nearby. But also, with limited dispersal, the ones that are nearby are more likely to be genetically similar to you. Okay? So you're more likely to produce a homozygote than you would if you mated at random across the whole species. Right? It's a kind of inbreeding. It's a very diffuse kind usually, right? It's not that you can say, oh, they're first cousins or they're second cousins or this is a nephew and this is an uncle or whatever. It doesn't work. Nephew and aunt. These are, but it's a kind of inbreeding where they may share one ancestor 15 generations back and another ancestor 10 generations back. So there's a complicated web of pedigree that causes individuals to be genetically similar, right? And that turns out to be measured by FST, the extent of that, that non-random mating caused by the fact that there's population structure. And here's just a simple mathematical cartoon to show you why it would have this effect. So this is, along this axis, is allele frequency. And then this is just showing you what would you expect the frequency of heterozygotes to be under Hardy-Weinberg, 2pq. Okay, so that's just a parabola. And so imagine that we took an individual, we took a population, population 1, and it had an allele frequency around, say, 0.2, whatever that is on this graph, and another population that had an allele frequency of around 3/4, whatever that is on this graph. So these 2 points would be the values of heterozygosity we'd expect to see within each of those populations. Okay. If we mix those together, right? If we took those populations and let them randomly create a new population that fits Hardy-Weinberg assumptions. They'd have allele frequency that was whatever this is here, about 0.45, so. And it would have a heterozygosity at Hardy-Weinberg equal to this. But the average of these two populations, on average, the heterozygosity would be the average of this and this, which is here. And that's the reduction in heterozygosity that we get because of the population structure. Now, this is called the Volland effect. And as far as I can tell, this is probably the least exciting thing to have named after someone in population genetics. But it's named after Volland. So it's the Volland effect. You mix two populations that are genetically different. If you mix them without allowing them to breed and create another generation, you'll see a deficit of heterozygotes. Okay. All right. But more to the point, in real populations, if we say, on average, what's the heterozygosity of individuals in those real populations, if there's heterogeneous gene frequencies across space, we'll have fewer heterozygotes than we would expect at Hardy-Weinberg. And they have fewer in proportion to FST. Okay. Let me walk through this a little bit here. I'm just going to show you a simple example of mixing two populations. And the same logic applies with more than two populations, but it's easier to see this way, I think. So as we have population 1, it's a Hardy-Weinberg within the population with allele frequency P1, P1 squared, 2P1Q, and Q1 squared. Population 2 has allele frequency P2. It's also a Hardy-Weinberg. Won't say him again. P2 squared, et cetera, right? When we mix those two together, right? If we just took that species and said, well, what are the gene frequencies, or what are the genotype frequencies in that species as a whole? It would be, if we mix these two populations equally, it'd be the average of those two. Okay, so P1 squared plus P2 squared over 2, et cetera. Now, if we're thinking about this as what genotype frequencies would we expect to see in a population that deviates from random mating, and describe the extent of that deviation by FST, just like we would with an inbreeding coefficient F, okay, but in this case we'll call it FST, we would expect to see P bar squared, P bar being the average of P1 and P2, P bar squared times 1 minus FST plus P bar FST. So that's just by definition in a sense. Let's say what's that, if we thought this was a measure of, if we wanted to find a measure of the reduction in heterozygosity and the increase of homozygotes caused by this kind of non-random mating, it should follow something like this. And it's going to turn out to be FST, so I went ahead and wrote it in there. So I'm cheating a little bit. I should have called that F who knows yet, right? And we'll find out later that it's FST. So if you then, right, so I'm going to equate this and this to say what would that FST be? It's on this slide. So okay, so here's the average homozygosity for the big A allele. Here's what we would expect it to be using this F as an indication of the change in genotype frequencies. Now we do a little trick here. So this, I'm going to write this as the expectation of P squared, right? Just the average of the P squares. And just doing some algebra here, moving around, collecting the FST terms, P bar minus P bar squared. Okay, now I'm going to move this P bar squared over to the left-hand side. So the expected value of P squared minus the mean of P quantity squared. Okay. Now, if you're used to working with the moments of distributions, you might recognize that the expected value of P squared minus the mean squared is actually the variance, right? Not quite by definition, but by one step away from the definition of variance. Okay, so this left-hand side is the variance in P. The right-hand side, we had left over FST P bar minus P bar squared. Well, if you factor out P bar minus P bar squared, you get P bar times 1 minus P bar Q bar. Okay, so when we get down to the bottom here, the variance in P bar, or sorry, variance in P is FST times P bar Q bar. Well, of course, that's just the same definition of FST that we already had, right? So that's the same number, the same quantity will predict the effects on homozygosity at the same time it's predicting the variance among populations. So that's kind of nice. Questions? I see. Right. Yeah. Oh, thank you. Yeah. Yes. Right. Right. So if you couldn't hear Deborah, what she was saying is this is not, this derivation was simplified, but this is not restricted to the assumption that the variants within that the individuals within populations are mating at random. So if you have inbreeding within populations, you can still describe this effect of FST, i.e., the reduction in heterozygosity caused by the population structure. That's still equivalent to this proportional amount of variance among populations. On top of which, you can describe with FIS what the amount of reduction in heterozygosity there is caused by that local inbreeding. It all still works. Did I phrase that? Mm-hmm. Yes. Thanks. Yes. Right. So if you couldn't hear that, she's saying when we're looking at genetic markers, we're sometimes interested in expected heterozygosity. So take a population that may have deviations from Hardy-Weinberg, but for that population, that local population, what would the expected heterozygosity be if it were at Hardy-Weinberg? And all these, what I've just shown you, pertains to that situation as well. Am I rephrasing that well enough? Okay. Okay, so yeah. Mm-hmm. Mm-hmm. Yeah. Right. So a good point. So I'll try to paraphrase the question and the answer. If you are looking at more than one locus with this kind of population structure and the allele frequencies vary at both loci, that can generate linkage disequilibrium in the population as well, just because of the subdivision. That's right. Yeah. So if by chance, I mean, and maybe tell me if this is not entirely what you're talking about, but if by chance a population, there's two populations, and one has a high frequency of big A and a high frequency of the big B at another locus, and whereas the other population had more small A's and small B's, at a population level, that's linkage disequilibrium. Yeah. And that can be generated by chance. If you have lots and lots of populations, you'll generate particular allele combinations, and on average, there'll be no LD, but in any particular case, there will be some. Okay. So okay, so that gives us a quantity that we might care about, something that we can use. I'll show you later ways in which we can use it, but for now, let's talk about this FST as a thing of interest. And I want to go back again to the roots of studying population structure. What I'm about to show you is something that is pedagogically pretty useful, but biologically pretty nuts. Right? So I'm going to show you a model which I have a real love-hate relationship with. Right? So if it's overused, it can cause you to be extremely wrong about your inference about nature, but I still think it's a great way to start thinking about the concepts involved. And that model is the so-called island model, mainly attributable to Sewell Wright. And the form in which I'll talk about it today, we have lots and lots of different populations in the kind of universal, those signs that work everywhere for bathroom or for stop sign, whatever. The universal sign for population in structured populations is the circle, and the universal sign for migration is arrow. So that's what this is. So these are populations. They're connected by migration. Now, with the island model and in its basic form, which often doesn't people don't often go beyond. In its basic form, each one of these populations is of effective size n. Okay, so you can think I probably should write NE, but we'll just call it n for short. It's effective size of the population, describing how much drift there is there. And M is the fraction of individuals that leave a population each generation. It's also the fraction of individuals which come into a population per generation. So if we say, after all that goes on, you look at all the individuals in a population and say, well, what fraction of these were born somewhere else? That's M. Okay? So technically, in this case, those two quantities are the same. They don't necessarily have to be in general, but in the island model they are. The two quantities that I just referred to are a backwards migration rate. Okay, so backwards migration rate, you say, okay, here's everybody. Movements ended. Where do they come from? What fraction came from somewhere else? The backwards migration rate is that. What fraction came from somewhere else? Looking backwards. The forward migration rate is here's a bunch of individuals. They're just about to leave. What fraction of them goes somewhere else? Okay. So those don't have to be the same for any given population, right? Yeah. No, it's n. No, it's n. The population size is not changing, it's n. Sorry? No, it's in the island model, it's n. You want it to be like biology. I applaud that. It's n. Yeah. We'll talk about that in a little bit. It's got those kind of deviations later. So it's n. N times m of them leave, and n times m come, and it turns back to n. There's a little minute while they're zooming around where it's something else, but every time we look at it, it's n. Is that the problem? Or you just didn't like it that it's not like biology? It's n. What? It's conservative migration, yeah. God made it that way. That's just n. Okay. So that's the island model. Now, I'm glad everybody's laughing at it because it's got a lot of problems. So like I said, it helps us learn some of the basic issues. So we'll go there first. So let's start. Sorry, we kind of already started this a bit. This chunk of the lecture, I want to talk about how drift causes differentiation in a balance with migration. Right? Okay. So the reason. Yeah, so the reason that for those of you at the tutorial yesterday that I chose that issue was because it's similar to this. We don't have to work through the whole thing as much. If you weren't there, well, you should have been. So we'll go through it. By the way, Brian wanted me to say that if you want to see more information about the issues we talked about in the tutorial, they're hidden somewhere in one of his handouts that are on the website. Your first lecture. It's population size handout. Finite population size handout by Brian. All human knowledge is, yeah. Right. So, but let's get back to population structure. So we're going to do we're going to do an infinite island model. Okay. So we're going to take the silly island model and make it sillier. We're going to assume that there's a lot of populations, effectively an infinite number of populations. Now that's not really a necessary assumption. It's just making that assumption makes it much easier to present this, and so it's good pedagogically. Okay, so because we're assuming an infinite number of islands, any individual which comes in is in no way between that assumption and the fact that individuals are moving independently to all the other islands, right? Hang on, I gotta go back because this is important. So here's the island model. Notice about this, another one of the creepy features of it is that when individuals leave, they go everywhere else with equal probability. Okay, so we're talking about space, but there's no space here except in some kind of folded fractal dimension of like 1/3 or something, right? So there's if you leave, you can go anywhere else with equal probability. Okay, so there's no isolation by distance, like the real world problem, but just be aware of it. Okay, so if you couple that assumption with the fact that there's of assuming there's a huge number of other islands, it means two things. It means that when migrants come to a population, they're completely genetically unrelated, right? We don't have to, and they're unrelated to the individuals which are there. So any excess probability of being the same genotype it isn't relevant to two individuals which come in, one individual which comes in relative to an individual that's there, right? Or for that matter, two individuals from the same population never go to the same place. Okay? So that means that gives, now look at this part's completely unnecessary mathematically. You can write two equations that track the probability of being the same in two different populations, the probability the same in the same population. It's just a lot simpler to make this assumption. So it's not a crucial one. It just simplifies things. And it simplifies it down to something very much like the infinite alleles mutation drift model that we talked about yesterday. Okay? So when an individual comes in, it's unrelated to the individuals which are there. It's novel. There's no longer any shared ancestry of those individuals. And so you get the same kind of equation. 1 over the 2 times the population size is the probability that two individuals share a parent allele in the previous generation. 1 minus that, the probability they have different parent alleles, in which case they were similar to each other in proportion to FST. But if those two individuals came from different places, then which happens with probability m each, then they'll be different again. So they have to come from the same place, 1 minus m squared, in order to be similar in the offspring generation. Now, there's a slight difference in the way I wrote this model to what we did yesterday, which is I have the 1 minus m squared inside rather than outside. You can get the other model just by changing the order of when you do population regulation versus when you do dispersal. So this model is written, we have dispersal and then population and then reproduction. If I pull the 1 minus m squared outside the whole right-hand side, that would be like having population regulation followed by migration, by mutation. Okay? And then it would look more like what we did yesterday. Okay. Now, we're going to make another glaring assumption. We're going to say, well, what's this like at equilibrium? Okay. So we'll do that the same way we did yesterday. I won't belabor it. And if you do, you wind up with this big equation, or the most famous way of seeing it is there in the bottom right, that FST in this island model with all these assumptions at equilibrium is 1 over 4nm plus 1. Okay. Now, that has lots of cool features that make people really want to use it. One of the features that it has is that we no longer have 2 parameters, we've only got 1, right? So we have nm in here. Nm has a biological interpretation. That's how many individuals, how many, not what fraction, but how many individuals move from one place to the next per generation. Right? That's kind of cool. That's something we might want to know. And in fact, people have used this to try to estimate FSD from genetics and solve for this number of dispersers. I'll talk about that more in a bit. Yeah. I'm not sure I quite heard. This is just right now. This is just migration. There is now. So if you add infinite alleles mutation to it, you get an equation that looks like this. Right? Again, an approximation. I throw this in because it's actually pretty relevant for a small subset of people, particularly doing landscape genetics with microsatellites and on things that are maybe quite distantly related, like something at a subspecies level or another. If you are trying to track what you expect FST to be, even if the island model was appropriate, and you thought that if you discounted the existence of that mutation rate, there are a lot of situations that people actually study, in particular when they start thinking about microsatellites, which have a very high mutation rate, where mu can be large relative to m. It could even be larger than m. like I said, microsatellites, subspecies, it's not going to be uncommon for mu to dominate the equation rather than m. And then you get completely misleading results if you're only thinking about this in terms of the dispersal dynamics of the species. Okay? That's a very, very common, unfortunately, very, very common problem. This equation, again, goes back to Wright, 1941 something. So it's not like we haven't known it. It's just that it's not been part of the way we think about these issues much. Okay. What questions? Yeah, so here's obviously just rearrange it. NM is a function of FST. FST is something we can measure, right? So it's not dependent on the specific average allele frequency. We can go out and find some markers that we think might be neutral, measure gene frequencies in different populations, calculate FST, and it's a very readily accessible quantity in a real population. So it's been very tempting to try to do this kind of inference. And I think one of the readings, I don't know how, was a paper we wrote a while ago now, describing some of the problems of that inference. And I want to talk through some of it, really not to bash this yet again, but because it gives a platform to think about deviations from the island model in the real world that are interesting. The island model makes a lot of assumptions. At root, in the previous slide, we're assuming there's no selection at the loci. We're looking at no mutation. All the populations are the same size. They all contribute equally to a pool of migrants. They all receive equally from that pool of migrants. There's no, and that in itself implies that migration is random with respect to distance. And then also, if you're using that previous equation, you're also assuming equilibrium, right? So that's basically, it's just saying, oh, let's pretend there's no actual biology for this organism, and then this will be fine. And that's not particularly what we want to do. The next slide, as I can see, but you can't, has nothing to do with what we're talking about. It's just in the wrong place. I'm just going to show it to you and talk about it, and then we'll regain our previous train of thought here. So, sorry about this. I'm just in the wrong place. I just wanted to show you that you can make similar calculations for FST, kind of another interpretation of, at least for an island model, in terms of coalescence, right? And so you can get to FST in a very straightforward calculation. You can say, what's the probability? You can say, if you take the individual, a pair of alleles in a population, so FST is about pairs of alleles. What's the difference? It's a pairwise comparison between individuals, essentially. So what's the probability? If you go backwards in time with coalescence, you go backwards in time, you can either, two things can happen as they go back in time for a neutral allele. Those two alleles can coalesce in this population where they are now. Right? They can both share an ancestor in the same place, or not, in which case that means that before they got a chance, I have a hard time with prepositions when I talk about coalescence. In the time that one of them has moved into that population since the time that they coalesced, since the time they had a common ancestor, in which case it would have been really, really long in the past because they were in different places and they had to, their paths had to find each other again, which would take a long time. Okay? So we can calculate an FST by saying which of those happens, what proportion of the time do individuals share an ancestor in a population since the last time they migrated. Okay? So the probability that one or the other migrated per generation is about 2m. Okay? Technically, it's 1 minus m squared, but that's about 2m. The probability that you had a coalescence in any generation in a population is 1 over 2n. Okay? So those are the two things, and those are the probabilities. What fraction of the time for a pair of alleles did they either the most recent event happen? Was it that one of them moved there, or that they coalesced? So the probability that its coalescence is more recent than migration is just 1 over 2n divided by 1 over 2n plus 2m. The two possible things are 1 over 2n, 2m. Those are the two probabilities. What fraction of that is 1 over 2n? Well, that's 1 over 4 and n plus 1 as well. Okay. And this is not mysterious. FST is actually about tracking the probability of two alleles being the same because they share common ancestors. That's why it predicts the variance. The variance is a pairwise comparison. That's why it predicts homozygosity, pairwise comparison. So it's the probability that two alleles are actually the same. Okay, is that a different way of thinking about it? Okay, back to our previously scheduled train of thought. Can we estimate, what are the problems of estimating dispersal by using these island model assumptions? Well, real populations are not like the island model. Very unusual to get a population that's actually like the island model. Populations vary over space. Migrations vary over space. They vary over time. Distance matters. Almost always, dispersal is more likely to be greater to nearby populations than to faraway populations. Right? Dispersal can be asymmetric. I don't know what I was thinking with this one. It's just kind of wild. But imagine a river, a series of salmon nesting sites along a river. I'm from BC, so we talk about salmon a lot. There's a salmon nest in particular stretches of the river where the stones are just the right size that they can be protected from the water flow, and those don't occur everywhere. So they're spatially separated up and down the river, separated by areas in between. So you might have, even in a river system, discrete populations. Individuals disperse between one generation to the next between those different breeding sites in a way that's asymmetric. They're more likely to not get quite as far as their ancestors did if they don't make it to the same place. So they're more likely to move from a upriver to downriver than vice versa. You can imagine the same thing if there's a waterfall on a particular river. It's true for guppies in Trinidad. There's waterfalls that separate upper and lower populations. There's a very strong asymmetric migration because guppies sometimes fall over the waterfall and mix and survive into the lower population. Guppies just cannot get up a waterfall to cause gene flow the other direction, right? Until Dave Resnick comes along and moves them, but that's different. Populations vary in their contribution to the next generation. This is a kind of subset of the differential migration, but some populations may be quite successful genetically or by selection and cause them to contribute more to other populations. That'll be more important tomorrow. And then a last one, actually I'll talk about it a bit because it was my first paper ever. I'll talk about it today. Sometimes populations go extinct, and new populations start by colonization, and that has a lot of genetic consequences as well. So let me just show you one slide about that. Here's the equation. I'm not going to walk through it at all. You can tell it's different from 1 over 4 and n plus 1. That's the point I want to make, right? There's other things that matter, like the extinction rate and the probability that individuals come from the same place and how big are colonizing populations, et cetera. If you took this, what this graph shows is if you took the FST from a population that had a fair amount of extinction and recolonization, you would estimate NM to be along this curve, and actually this was the actual, it'd be very, very far off. This is, I think, assuming an extinction rate of 10%, which is on the high end, but just to make the point. Okay. One last assumption that I want to talk about is this equilibrium assumption, and this is just a simple plot for a single case where population size is 100 and migration rate is 1 in 100. At equilibrium, that goes to approximately 0.2, regular island model, except we're starting it out of equilibrium from a well-mixed population, and it takes a while to get there. So these are 50, 100, 150, 200 generations on this axis. These times slow way down if you have large populations and weak migration. Okay. But if you have small populations or high migration, it goes relatively quickly. This is relatively quickly. Right? So, but it can be, if you measure things at a particular point in time, when you're starting out of equilibrium, you can get very wrong answers. So populations of trees in North America have only been there for like 30 to 60 generations since the last, northern North America, have only been there for 30 or 60 generations perhaps since the last glaciation. If large population sizes, we wouldn't expect them to be at equilibrium. Maybe some species of conservation concern, large animals with small population sizes may have gone from high uniformity to a situation that will eventually cause a lot of genetic differentiation, but it may not be there yet. Okay. So let's not use the island model per se. There's a lot of issues there. Okay. Did I start an hour ago? Is that right? Yeah. Yes. I'm sorry, I couldn't hear that. Yes. Yeah. So this slide, I assume that the demographics of the population weren't changing from this point on. There's populations, each one size 100. There's migration equal to 0.01. That stays the same all through this. That's not changing. It's just that the FST is not at equilibrium at the beginning. It starts at 0 in this case, and it doesn't instantly go up to 0.2. It has to work its way there. Yeah. The drift has to accumulate those differences over time. So there are some more sophisticated approaches that are more commonly used now. They still make a lot of assumptions, and I don't really want to, I don't have time to go into them right now. A program by Peter Beerley called Migrate, by a program by Jody Hay and Rasmus Nilsson, right? Called IM. Is that right, John? Yeah. So these make a lot of assumptions themselves. They're better than just going from the island model like I just talked about. What I want to encourage you to do, and this will be a side note, and nothing on this slide other than pause to talk about this. What I want you to do is when you have genetic data and you're trying to infer something about it using some established technique, it's really, really important to learn what the assumptions are of that technique. And I'm sorry to say that those assumptions are not always clearly stated by the authors of those methods. And sometimes they're more clearly stated early than later, and they start assuming that people know them, or they start thinking that, oh, this is the way biology works because this is the way it works in my model. And people can make a lot of false inference based on methods that don't have assumptions that fit the biology of the organism they're studying. Now, no method will ever perfectly fit the biology, and there's going to be deviations that don't matter. But there are real important deviations between what's assumed by the models, by the approaches, and by real biology, and it's really important to be aware of what assumptions are being made and to try to think about how those matter in terms of what you're seeing. And I know that's stating the obvious, but it's a really challenging thing because we don't do a very good job as a field of exploring the effects of these assumptions. So there's not always a huge literature to tell you what the effects of those assumptions are. So just be cautious in using these population genetic approaches. They can tell you a lot, but not necessarily to the level of precision or accuracy that you might hope. All right. So in the last few minutes, let me pause for questions and I'll switch gears. Okay. So I want to switch gears again to the other source of differentiation, the other major source of differentiation between populations, and that's selection, spatially heterogeneous selection. Selection varies over space. Again, that's a feature of everything. Whether or not that's strong enough selection to cause patterns is what we want to explore. Selection varies over space because the environments vary over space. That spatially heterogeneous selection tends to increase genetic variance among populations, right? It tends to cause alleles which are favored in a place to be at high frequency, and the alleles that are favored somewhere else to be a high frequency there. And those may be different, and that propagates genetic variation. And migration, of course, reduces that. So, so I used this term on the previous slide, local adaptation. This is something that, in principle, has an easy meaning, and in practice can be a little bit difficult. Local adaptation is when the mean fitness of a local population or a local individual exceeds the mean fitness in the same place of individuals from somewhere else. Okay? So if you grow something where they come from compared in the same place where they come from, compared to something from somewhere else. If the local ones do better, it's evidence for local adaptation. Okay. Let me quickly show you just a meta-analysis that was done by Liumu and Fisher a few years ago. These are on reciprocal transplant experiments. It's a very nice experiment to try to measure, style of experiment, try to measure local adaptation. Here's a cartoon of the basics of the experimental design. You take individuals from one population, they're different colors to try to reflect maybe their different environments. Take individuals from here and plant them here and there. Take individuals from here and plant them here and there. I say plant because these experiments tend to be plants. You can in principle do them with animals. And then you can compare the fitness of local to foreign individuals reciprocally. Okay? So they're transplanting, and you're doing it reciprocally, reciprocal transplant. So here's based on over 1,000, really a lot. I think over 1,000 reciprocal transplant studies. And here's what they found, and there's a really interesting angle to this that I'll come back to in a bit. So these are positive. That meant that both transplants showed evidence of the local one being better. Okay. This is positive, negative. One showed evidence of the local thing being better, and the other, the wrong one, won. And this is negative, negative, where the foreign one was better in both places. Okay, so you can see there's a nice preponderance for some kind of local adaptation. The distinction here between the gray and the white bars is these are studies where the population size was what they called small, so less than 1,000 was their criterion. And these are studies where the population size was large, greater than 1,000. And so we'll come back to it in a bit, but you can imagine that you'd expect selection to be more effective in a larger population. And in fact, that's what they showed. You get more evidence for local adaptation in larger populations. So you get a lot of local adaptation. You don't always. So let's go back to the very simplest model we can think about with local adaptation. And that's a continent-island model. So imagine there's a continent, this giant yellow blob down here, and an island over there in blue. To keep our lives very, very simple, we'll assume that the continent is unaffected by the island. There's no migration coming from the island to the continent. In the continent, you want to be big A. That's selected for everybody's big A on the continent. And there's no migration back from the island, so there's nothing keeping it from being what it wants to be. It's basically big A, 100% in the continent. The island, a little a allele is better, right? So over here, a different allele, small a, we'll call it, is favored. And we'll say that the fitness of that, do a haploid model, fitness of that is 1 plus S on the island. And migration from the continent to the island at rate m per generation. And that m is the backward migration rate of the island. Okay. Now, so the allele frequency of small a is 0 on the continent, just fiat by assumption. What is it on the island? What's it going to be like? Well, so we can do this in a very, much nicer way, but in a very, let me just show you in a very straightforward way. The change in allele frequency of that small a allele on the island is minus mq. You can think about that as for any individual which is a small a, there's a chance that it gets replaced by a migration event that happens m of the time, in which case that small a allele goes away and turns into a big A. So minus mq is the change in the little a allele frequency due to migration. And the effect of selection is approximately SPQ. So the change in Q by selection is SPQ. Right? This is I don't think we've ever done it quite in this simple way, but with haploid selection, weak selection, you can get this simple approximation for the change in allele frequency, SPQ. So the overall change in allele frequency is the change due to migration, the change due to selection. And that allele frequency will be greater than 0. The little a will be greater than 0. So there will be some local adaptation when S is greater than M at this level of approximation. It's very intuitive, very, very sweet, intuitive result that you'll get some local adaptation. The little a allele will be able to succeed at some extent simply if selection outweighs migration. Yeah. Yeah, it got there to start with. Let's see it got there because it mutated on the continent and moved in, or mutated on the island. It's there. We're not thinking about mutation yet. Just will it stay there once it's there? I guess is what we're after here. Yeah. Okay. So it's just can we preserve that variation? Well, selection has to be stronger than migration. Right. So that could be that the small allele becomes quite frequent. If S is much greater than M, then you would reach something like a migration selection balance, the dynamics of which would be similar to what we talked about 2 days ago with mutation selection balance. Okay. So let's make it a little more complicated. Bulmer in 1972 did a model of 2 populations. And I apologize, I'm going to actually change the variables up through this. Now you've got 2-way migration. The 2 populations are in the way I'm about to show you, equal in size. One allele is favored here and relative to the other allele, and disfavored here relative to the other allele. So there's local, spatially heterogeneous selection, right? So one allele is favored one place, the other in the other. And Bulmer showed that you can maintain both alleles, i.e. you can maintain variation under this, this conclusion, right? M1 over S1 minus M2 over S2. This is let me just show you that graphically. It's a very strange thing. So this is the area where essentially anything inside these zones will be where these get deterministically maintained in these populations. Okay. It's a very strange shape. So when you get very close to symmetry of the migration rates and the selection rates, you can have very, very weak selection and still, at least in this deterministic model, maintain that variation. Okay? But it tends to be the case. Right, I'll show you that. Sam Yeaman and Saliato followed up on that model, and mainly the reason I showed you that model was to show you this one. This is essentially the same model as before. I just cut and pasted this out of their paper, which is in Evolution in 2012. So this is written in terms of the fitnesses rather than the selection coefficients. But a thing that appears here that wasn't there before is the effective population size. So as the effective population size becomes small, it becomes harder to maintain this variation between populations. Right? And the core insight of this paper, and the reason I wanted to share it with you, is that the dynamics of allele frequency in these 2 population cases, we've heard a lot about the probability of invasion of alleles, et cetera, being a function of S. In these cases, it is a function of S, but not uniquely. It's a function of essentially the difference between selection and migration, right? So the force increasing the frequency of an allele in the place that it's favored is a function of S, but that's counterbalanced by migration from the other population. So the total deterministic force changing the frequency of that allele is a function of both S positively and M negatively. So that leads to this case where it doesn't take nearly as much drift as you would imagine to make the deterministic prediction not work, right? Because it's not about how strong selection is, it's about how strong the difference between selection and migration is, essentially. Okay, and so the eigenvalue, the eigenvalue minus 1 of the dynamics of this is what determines the invasion probability in either direction in these models. And so drift becomes really important. And if we go back, this is from Yeaman and Otto, with smaller and smaller population sizes, you get smaller and smaller zones where you can actually maintain that variation. Okay. So drift matters too. And so tying that back to the results from the meta-analysis of reciprocal transplants, remember what we saw there was what they called small populations, less than 1,000, were much less effective at maintaining local adaptation than the larger populations. And I think the reason's probably related to this. Now I want to jump a little bit up from population genetics here to still talk about this issue of what maintains variation in spatially heterogeneous environments. This is a very old question, and one of the key insights from the 1950s was that a lot of population genetic models make assumptions that the population size is constant, right? That's not necessarily true when you're talking about issues relating to local adaptation. With local adaptation, you can improve the fit of a population to its environment, and you might actually increase the success of that at the population level in that environment, right? So in particular, if we have something that's come to be called, unfortunately, been called soft selection, with soft selection implies that individuals compete only against other local individuals for a share of the next generation. Okay, so you think of that simply as saying that population regulation is local, not global. Right? So if an individual somewhere does well, that may decrease the resources available for other individuals locally, but has no effect on somebody somewhere else. Right? That's soft selection. Something like that must be happening often. Hard selection is when there's equal competition among all pairs of individuals regardless of where they are. With soft selection, actually reduces the efficacy of global individual selection. Right? You're no longer comparing individuals from here to individuals over there directly. Right? You are in the longer term as things move around, but not on a per generation basis. And so, in effect, it shrinks the effectiveness of selection, right? Because you're not competing against the same range of alternatives. And so you can more easily protect polymorphisms. You can more easily maintain genetic differences with soft selection. Okay? So something that does well, if something does well here, something else does well there. If you have soft selection, it's very likely that you'll be able to maintain that variation. Independent of M, so here's an equation that actually assumes complete migration every generation due to Levine, 1950-something. E1 and E2, as I've written it here, are the proportion of the population that are in different environments. These are the relative fitnesses of the A type relative to the little a type in population 1 or population 2. And the similar equation over there, if both of those are true, which you would get when you have a good fit of each individual to its population, you maintain polymorphism. And it actually, migration reduces the rate of that substantially, reduces the differences substantially, but it is always re-perpetuated each generation via selection. Okay. So you can see this polymorphism even with quite high migration rates. And you were asking before about what if individuals move non-randomly with respect to what's good for them. Again, it's called habitat selection. That's also a major mechanism when it happens to create variation among populations. So the individuals themselves sort themselves out by where they should be, and they tend to take genes that are good for that local environment to that local environment. So habitat selection greatly increases local adaptation, certainly greatly increases the zone of cases where you might get local adaptation. Okay. I just want to show you one more slide before I do. Anybody got any questions about? Yes, yeah. Well, I think if you had habitat, they didn't do habitat selection. If you'd add habitat selection to this, my guess is that would that could potentially be a strong enough effect that the drift wouldn't matter. Right. So if you would add on top of the selection and migration function another force that would cause the association between genotype and environment, and if that were stronger than the S minus M, it's not really S minus M, but that kind of thing. If that were stronger, that could take you out of the zone where, yeah, you're adding another deterministic force on top of this, and if that were stronger than 1 over the effective population size, that would be enough to maintain the difference. Yeah. Right, right, right. Oh, well, one thing that leads to asymmetry is when you have large populations relative to small populations, and so you have asymmetrical gene flow. The large population is likely to be well adapted, and it's going to be sending locally maladapted alleles to the other population, for example, right? I mean, there's lots of dynamics that go into these when you start breaking the symmetry. Yeah. What was I going to say about that? Yeah, and there's a great school of thought that Dick Gamakiewicz, who will be here on talking on Monday, I guess. I don't know if he's going to talk about it, but he's done a lot about how does a population adapt to a new environment when you have a single large population that's occasionally sending migrants into that new habitat, and initially they're maladapted and can't persist, can't reach a fitness great enough to replace themselves. But what are the dynamics of that? And it's some really cool stuff. It turns out that an intermediate migration rate is optimal for being able to colonize that new environment, because you need enough individuals to get there to be able to try it out, to have a high enough probability that eventually a mutation lands there that's good enough to succeed. But if you have too much migration, then it swamps out that and is continually replacing that locally adapted genotype with the one from the other. There's some really cool stuff, and you should talk to Dick about it. It's good. Okay, so the one last slide I want to show you, oh, that's the wrong way, is this one. So when we think about the genetics of adaptation, and we think about what kind of, how big should we expect the alleles that we see causing adaptation? Now, so Alan Orr has done a lot about this, as well as other people. And Alan proposed that the distribution of allelic effects causing local adaptation should follow something like an exponential distribution, something very close to an exponential distribution. In other words, the most frequent alleles contributing to adaptation should be weak effects, but there'd be some distribution out to large effects. Right? There's been later estimation procedures based on that. And I just want to finish by saying that one key insight that we can get when we have discrete populations like this, we've just seen, if on the second part of the talk here, if there's one thing that you took away from, I would hope it would be this, that if migration exceeds selection, with maybe some coefficient on one or the other of those, depending on the details, but if migration is large relative to selection, you're not going to be able to get local adaptation, usually. If selection is strong relative to migration, you can. Okay? So that makes a prediction about the genetics of local adaptation. Right? That predicts that for local adaptation, we should see a preponderance of alleles responsible for that local adaptation being a relatively large effect. And the weak effect ones, if they're there, may not be there because they generated local adaptation. They may just be there for some other reason. Right? So here's a graph done by Cort Griswold from 8 years ago, where he, by simulation, had a distribution of allelic effects, two populations with migration between them, and then asked, of the alleles which differentiated between the populations that caused them to be different, that created that local adaptation, what was the distribution of those alleles, of their effects? And this is what he found. He's found both positive and negative directions. He was combining the alleles in either population in both directions, but there's a dip around 0, right? So the alleles of weak effect here at 0 were not contributing to local adaptation. They were not differentiating. They were overwhelmed by migration. So as a very practical matter, we should expect, if you're looking at QTL studies or some other way of trying to find the genes responsible for local adaptation, we should expect that to behave differently. The pattern of genetic variation that we see there should be different than what we might expect for, say, disease alleles, which may be more like mutation selection balance, and have lots of low-frequency alleles of all size. Right? It should be different from the pattern of adaptation that we'd see from a directional selection in a single population, where, as Alan Orr showed, to be more exponential. The good news about this is the ones that we can see with our methods, right, with anything like a reasonable sample size, the ones we can see are the ones above some threshold of magnitude. Right? So I'm suggesting that for local adaptation, we may be able to see a large fraction of, in certain circumstances, when you have this kind of discrete environment, you may be able to see a large fraction of the alleles responsible for local adaptation, and they should be easier to find. And we certainly shouldn't extrapolate back by some exponential to assume how many are down in this part that we can't see. That's been done before. It's not probably a good idea. Any questions? Yeah. Yes. Right. Right. Yeah. So in the case of drift, yes, for sure. It's largely proportional to the effective size of those populations. Oh, yeah. Yeah. You mean growing and shrinking as a function of? Right. Right. I don't think that I don't know of anything that's explicitly followed, say, in chaotic dynamics or any particular dynamic. There's some work that's been done that just says, well, if we just knew the variation of population size, how does that affect? So if you had white noise in population size, yeah, in the mean population, no, white noise in the local population size. What does that do to differentiation? It tends to increase differentiation because it, as I think Brian talked about, that tends to reduce the effective population size. It's a lot like the extinction colonization model with the white noise. Is when the populations get small, they have a lot of drift, and they have a lot more drift when they're small than they have less drift when they're large, and so that leads to more drift in general. I don't know of any. There must be work, but I can't think of it right at the moment about how local adaptation is affected by changing population dynamics. Okay, Bob Holt, she says. Yep. Right. Okay. Let's eat.